For those familiar with GCOOS, we're one of 11 regions of the U.S. Integrated Ocean Observing System. Our mission is to provide on-demand information for the Gulf of Mexico. We have 4 main focus areas where we concentrate our efforts. That's marine operations, coastal hazards, healthy ecosystems and living resources, and human health and safety. And then we also have 4 cross-cutting themes. And those are outreach and education, data management, and we are no longer a data management and communication. IUS has gone to data management and cyber infrastructure, and numerical modeling and forecasting, and monitoring long-term environmental change. And so the real reason we're here is to hear Dr. Dan Otis, talk about the Ocean Data Dashboard. This was developed to enable a better understanding of historical and current changes in ocean conditions. The webinar is being recorded and it's going to be posted to the GCOOS YouTube channel. And please remain in listen-only mode. If you have questions in the chat box, we're going to wait until the end to answer the questions unless there's anything critical that's really going to impact the understanding of what Dan is saying. So I'm excited to welcome our speaker, Dr. Dan Otis. Dan is an oceanographer at the University of South Florida College of Marine Science. As part of the Institute for Marine Remote Sensing, his research activities focus on the analysis of digital data obtained by satellite and airborne sensors and on the development of applications of these data. And his talk today is going to showcase the structure of the Ocean Data Dashboard and provide case studies of how the information is being applied. And the work is contributing valuable lessons right now to how to best incorporate biological data from the Marine Biodiversity Observation Network into the IUS enterprise. It's really a nascent part of the program ramping up, and so these kinds of exercises are really important in setting the stage for future work. So Dan, all yours.
Yes, okay. Thank you, Chris. Thanks everyone for joining today. I'm Dan Otis. I work at the USF. So these are just a few partners here that are part of this whole enterprise that we've been going about 4 years now. So I will jump right in. Let's see, there we go. So today, let's see, just a brief outline here. Going to talk a bit about motivation, how this all got started, and some of the goals that we have in terms of what sort of phenomenon we want to try to capture with these data dashboards. And then dive in a little bit to the dashboard structure, talk about what you'll see when you go there, some different ways you can search for data and change the time period, change the location where you might be able to look at extracted data. And then we'll look at a few case studies about interesting phenomenon that you can see in the data that's presented here, that's displayed. And then just a tiny bit about future plans, where we're headed, some things that we would like to try to incorporate in the future. And then depending on if there's time, I can show a live demo of one of the boards and probably take any questions at that time. So let's get started. The motivation here, it all started back in 2016 when some of our colleagues at the Flower Garden Banks National Marine Sanctuary had some reports of a highly localized coral and invertebrate mortality event that struck on July 25th, or that was the first report of it in any case, in 2016. And so, some things came out of that event. There, if you look here, the Flower Garden Banks National Marine Sanctuary is in the northwest corner of the Gulf of Mexico, and the staff for the sanctuary is based in Galveston, which is some distance away. So if they want to plan trips to monitor the reefs, they need to go on about a 10 or 11-hour boat ride. So it's not something they undertake lightly. And so, in talking about this event, and we responded with some imagery, and they had some interest in wanting to know about conditions in pretty near real time, kind of in preparation for when they were going to head out, what were things looking like, and so we did that. But then that kind of gave me an idea that, well, we could automate some of this, so we could we could have a website that people could go to and see data for their region, mostly satellite-based data, but also things that would contribute maybe to conditions that would impact the reefs. And in this case, we wrote a paper on this with Matthew Lahanoff, and Michelle Johnston also wrote a paper about this event in 2016, and we saw this later in the imagery, and I'll show this as one of the case studies, but basically this big band of high nutrient-rich water built up along the coastline, and it was due to a period of high rainfall, which led to high river discharge, both from the Mississippi and from 5 rivers in Texas. And so the idea was that we could try to catch some of these things to observe the discharge increasing and look at what was happening in the coastal areas, and then try to set up a system where we could kind of foresee if this nutrient-rich waters were making it offshore and might impact the reefs. So that's kind of how it all got started. We had a workshop that happened there, and a lot of people got together. And basically, we in the satellite image, we saw that these high nutrient waters, rich in organic matter, made it offshore and impacted the reefs, and particularly impacted the East Flower Garden Bank location. There's 3. There's East Flower Garden Bank, West Flower Garden Bank, and Stetson Bank here, just a little bit to the west. So that's kind of how we got started. And so working with Frank Miller-Carter and Tyler Murray in our lab, we kind of set to design a system, and it's evolved over time. But basically our goals were to provide a one-stop shop where, for a certain region, data managers or other scientists or even members of the public could go to this website and see what was happening, not only currently, but also could go back in time and look at, try to get some context for what was being observed right now. So a lot of the things that we do here are based on anomalies. So in other words, for the satellite imagery and the time series, we basically calculate a climatological mean using a set of the data, in this case from 2013 to 2019, and we subtract that on a weekly basis. So basically we end up with, rather than just the raw chlorophyll number or the temperature, it gives you some context into how what the measurements are now are relating to the historical patterns. So that was one of the goals, was to provide this this interface that people could use relatively easily without having to go to a bunch of different places for this data. And we wanted to keep it in near real time, and we also wanted to go back as far as the satellite record would take us, which in this case is 2002. That was for the MODIS sensor. We also wanted it to automatically update, and this is kind of taken a little bit. I didn't want to have to update it manually every week or every night. So we've kind of, over time, we've streamlined this process. And then through the interface that we use, basically we wanted to set up a system where we could put in a threshold, let's say for a certain parameter, say if sea surface temperature went above a certain threshold, that it would generate an email automatically that would alert someone, whoever was interested in that particular parameter at that location, that conditions were departing from normal. And we also wanted the ability to add more data sets over time, which we have. We're looking to take more steps into the future to add in situ water quality data, perhaps some biological data. And so we wanted an interface that was somewhat flexible, and that's sort of a work in progress at this point, but definitely that's where we want to be able to have the ability to add more data over time without having to reinvent the wheel. So, and some of the phenomena that we wanted to capture, it started with these nutrient-rich waters in the Flower Garden Banks region. Were those nutrient-rich waters going to make it offshore, impact the coral reefs. On the West Florida shelf, we had a big HAB year in 2018. I'll show a little bit about that later. We wanted to not only include satellite data, but we also wanted to include other in situ data that we could capture without having to, do too much heavy lifting ourselves. So we basically, tap into data sources. River discharge comes from the USGS, and in the Keys we have some in situ buoy data that's run by the National Park Service. In the Everglades in 2015 and 2016, there was basically, we had a really dry summer in 2015, and it led to a lack of discharge into Florida Bay. And if you recall, we had some very high temperature, hypersaline waters that led to quite a bit of seagrass die-off in the Florida Bay. So we wanted to be able to capture events like that. We wanted to be able to capture, tend to be short-lived, but events where, sediments are resuspended by storms. And we can catch that with one of our turbidity products. We also include temperature products that can capture coral bleaching. And recently, working with partners at the Florida Fish and Wildlife, we have added some coral disease observations, and this is in progress. We're finding different ways to visualize these trends. The stony coral tissue loss disease has propagated pretty much throughout the entire reef tract, starting in 2014. So one of our dashboards is based on the needs of that community and how we can basically extract data in places of interest for them, and also display some of their data, which is basically observations of the disease or observations of absence of the disease at different locations along the reef tract. So as it is now, we have 3 dashboards. We started with the one in Flower Garden Banks. That was in 2017. We had sort of a somewhat crude interface at that time where I was generating pictures of each satellite image. We've since gotten a bit more sophisticated. In 2018, we basically made a version of it for the Florida Keys National Marine Sanctuary. And then just earlier, late 2019, early 2020, we've made the one I just mentioned, which is funded by the DEP and working with Luke McCacron at the FWC to incorporate basically a version of the Florida Keys dashboard that extends a bit farther north and incorporates the observations of coral disease. So that's kind of where we're at now. We're working currently to update our back end with Tyler Murray, who works in our lab here at USF, and we're basically packaging and getting things ready with the goal to transfer the hosting of these products to GCOOS. That's something they're really good at, and they host a lot of data for the Gulf. So we're looking forward to turning that part of it, working with them to host these products and hopefully get some more people using them. So in terms of the products we have, it's, primarily we rely on satellite data from the Otis satellite, and also more recently the VIIRS sensor. These Otis has been up since 2002. It's reaching the end of its lifespan, but it's really been a workhorse for ocean color sensors. It's been going for 18 years now. And recently, a follow-on to it, or an extension, is the VIIRS mission, which measures most of the same things. And so that one should be going farther into the future. Of course, there's always new sensors coming up, but these are the 2 that we are using right now for our imagery. And so the satellite imagery that you'll see on the dashboard is binned into 7-day composites. Daily images tend to be contaminated with cloud cover often. So we found that if we put together 7-day images, basically averaged over a week, that you can see patterns and get pretty good looking images. And in addition to that, we extract time series from those images, actually from the daily images, and we put those on the dashboard as well. And I'll show you some of that coming up. So the source, we get our satellite data via subscription from the Ocean Biology Active Archive System. That's at Goddard Space Flight Center. And so the primary products we use are chlorophyll A concentration, which is a proxy or an estimate of biomass, phytoplankton biomass, sea surface temperature, harmful algal bloom. We have a product called the Algal Bloom Index, and it's based on the normalized fluorescent line height product. That's basically a way to discern phytoplankton blooms from chlorophyll A concentration. And it's basically the fluorescent line height product, and it's normalized for suspended sediment. So we wanted to try to not have false positives from suspended sediments when we're trying to look at HABs. And then to look at turbidity or suspended sediments, we have a red reflectance product, basically at 667 nanometers. And it's basically just looking at the very surface. And it's a product that will tell you when you have a high level of suspended sediments in the water. And so basically, like I mentioned earlier, we calculate anomalies from these products. And we basically do that by subtracting the climatological values on a weekly basis. And we use data from a 7-year span from 2013 to 2019. And in addition to the satellite data, we have other environmental data. The USGS provides daily river discharge data from many locations throughout the country. It's really easily accessible. It's basically, we can grab it from the internet each night, and we just simply add it to a time series. And yeah, it's really nice to work with, and it's they don't actually go and observe it. They're gauging stations. But it's most rivers in Florida, for a lot of them, have been monitored since the 50s, or in the Keys since the early 2000s. The National Park Service also runs a series of buoys in Florida Bay, and those are basically give us in situ temperature and salinity. And these are nice because you get a temperature record that's not subject to cloud cover, so you get it, and their sensors are about half a meter below the surface. And you also get salinity, which is nice, and that ties in with river discharge, and it's a product that we can't really get from the satellite. So it's nice to have that. And then just recently, we've added coral disease observations from FWC, Moat, and others. They have a set of several data sets that they're currently sort of updating and curating, and we're going to get that onto it's on there now, but we're going to make it easier to see, and we're going to make some nice animations of how the disease progressed throughout the reef tract. And so basically, those observations are, when they go out and monitor this, do they see the stony coral tissue loss disease, or if they don't see it, they mark that down as well. So that's on the latest version, the FWC dashboard. So this is sort of a busy slide. Don't worry about this too much. This is just the data flow, and as you can see, there's quite a bit that goes on behind the scenes. And that's fine. We've streamlined this process over time. Our images are stored on ERDAP servers, which makes it easy to share with other partners. Time series data is saved as CSV files, and we've made those available so people can download those if they'd like. The processing routines run automatically at night. And for the front end interface, what you'll see is an open source software package called Grafana, which was designed to monitor servers, computer servers, and then the images are served via ERDAP, and then Grafana actually pulls those images in and you can grab them as well in a picture form. So let's jump right in. So the dashboard structure, basically at the top of each dashboard has a map. This is the one for the flower garden banks. And so you'll see here the things of interest are in red. We have the three reef locations. And so basically the locations that you see here are called out. And the reason they're on here is because these are locations where you can extract, you can visualize extracted data. So for example, at either of these reef locations, you can look at the extracted time series, or these are what we call our sentinel sites, these numbered yellow dots. And basically these were designed to, if you look at the chlorophyll record, say, and you have a big pulse of chlorophyll-rich water coming from say the Mississippi from the east, you're going to see it at these locations before it's going to impact the reefs. And even before that, you're going to see them in these coastal ROIs, regions of interest here. So these have been added to basically try to catch things that are happening that might lead to anomalous conditions before they make it all the way out here to the reef locations. And then the rivers you see here, the rivers, the green dots are where the gauging stations are for each river. And so those are where we get the data from the USGS. And so that is for the flower garden banks. And I think next, this is the the Florida Keys. This is the marine sanctuary is that border, is this red line here. So that's the outline of that. And then we have a set of these yellow dots. In this case are the buoys run by the National Park Service. And here we have 5 rivers. And these basically are turned into an index, and we sum these to make an index of discharge from the Everglades into the Florida Bay and the eastern part of the West Florida Shelf down here. And then we have a set of ROIs. These are a little different in that they're polygons. So this was based on some discussions with Andy Bruckner at the Florida Keys Sanctuary and Kelly Montanaro at AML, and she's also part of the Florida Keys Integrated Ecosystem Assessment. And they were interested in looking at different ROIs, different regions along the coast, along the reef tract within the boundary of the sanctuary, and then some other ones as well, places where you're likely to see water cutting through, like this is a big area where water from Florida Bay will make it out onto the reef tract. This East Florida Bay location as well. The Shark River empties in here. This is a very murky, turbid area. And as you'll see too, a lot of the, particularly a lot of things coming down, like the harmful algal blooms in 2018 will kind of make it through this region. They'll come down the shelf, and if they're going to make it out onto the reef tract, they're going to kind of make it down here. And so we put a region of interest here as well. And so that is the Florida Keys. And lastly, we have the Florida Fish and Wildlife Dashboard, which is, as you can see, it's similar to the Keys, and it extends a bit farther north. And we have some 3 other rivers that we've brought in. This is the 135th Street Canal in Miami. This is the Loxahatchee River and the St. Lucie River. And then we have our same 5 rivers on the west side of the state. We have our same buoys. And in this case, the interest on the part of FWC was to extract data at these 8 locations. And you can see they're called out here. And so oftentimes, if you'll see, after periods of high rain, that you'll get a noticeable signal in the chlorophyll here, say at the St. Lucie Inlet. So there definitely is a link that we've discovered or that we've observed with these data, that you can see a close link with some time lag between periods of heavy rainfall, an increased gauge height, and then you'll see a chlorophyll signal here, or in Miami or other places as well. So that's basically the top of the dashboard. It's kind of orients you to where you're at, and that's how they all kind of have that form. And then also at the top of the dashboard, there's some choices that the user can make. The time series and the set of imagery that you can look at is basically all confined or constrained to a certain time period that you can select. And so these, here on the right, you can go anywhere from 6 hours, which really wouldn't be very meaningful in our case, but you could choose between 5 years, 2 years, or you could also actually set, if you're going to say look at a certain period of say 6 weeks or 8 weeks, you could manually set that in these two boxes. Likewise, you could choose to look at extracted data from any of the 8 locations that you saw in the map in the last slide. You can likewise choose which buoy that you wanted to see data from here. And you can choose multiples if you wanted to say compare what was happening at Sugarloaf Key versus Sombrero Key. You could select those two, and then your plots would show that. If you select a lot of them, the plotting tends to get a little busy, but you can definitely see when certain locations have high values, higher than other locations. So that's kind of at the top, and the user will set that, and that will basically take you where you want to go in terms of time and location. And so then below that, you'll see the panels of satellite imagery. And like I mentioned before, these are weekly images, and they're anomalies. So we're looking at, we've basically subtracted the climatological mean. Here we have chlorophyll, we have sea surface temperature, and this is our algal bloom index product. This is mostly quiet, and then you see when there is a bloom, you definitely see a strong signal here. And likewise for the red reflectance, many images are green, which means that it's basically at a 0 anomaly. So it's, conditions are normal. But then every now and then when you have most of the time in these cases so it's driven by the high signals in this product are driven by a wind-driven resuspension. And so you'll see these, they're often very short-lived. Like you can see here, there's quite a bit of, close to the coast. Because if you were looking at an image of this, or say chlorophyll, and you were just looking at the mean value, because it's so shallow, you would see a lot of red colors, and it would be hard to discern. If you had, say, a pulse of something close to shore, you wouldn't necessarily see that. And that's why we choose to look at anomalies for display here in these products. For the FWC board, we have these coral disease observations. So this is % of sampling sites affected. So basically where they saw the disease, and then these panels down here break it down by year. So this is 2014 through 2017. And so these are a little hard to see, I'm sure, but you can see that in the red is where the disease was observed, and the white was where they made observations, but didn't see any of the disease. So this is a little bit static, and we're working to make some animations that will hopefully, be a little more illustrative in terms of how this disease made it down the reef tract. And then below that, we have the satellite time series, which are extracted from the locations that that the user selects at the top. And so in this case, we have sea surface temperature at the Miami location. The next one would be chlorophyll A. We have the algal bloom index, the turbidity proxy, the red reflectance. And then we have one that I didn't mention, which is basically the diffuse attenuation coefficient at 490 nanometers. That's in the green, and it's basically a proxy for water clarity. And here you might not be able to see it really clearly, but the yellow line is the mean, and the green line is the climatology. And so you have choices for that in terms, you could display these two together, or you could display either one by themselves, or you could instead look at the anomaly. And so that gives you some flexibility in terms of what metrics you might like to see and that's once again selected at the top. So, and then below that is the river discharge. And so basically we see here, this is for the East Coast. Those are those three rivers with the red dots in the map I showed earlier. And then below that is the green dots. This is a sum of those 5 rivers. You can see here, this is a fall of 2017. So we had a high pulse of discharge from Hurricane Irma, and I'll talk more about that in a second. And then finally, we have the Insitu buoy data. This is in the Florida Keys. And you can see here, once again, we have the mean as a yellow line and the climatology as a green line. We have salinity here on the left and temperature on the right. And then at the bottom, we just have some information on data sources if you want to link to NASA's, the sources of the satellite imagery or the USGS data or the buoy data. That's all here. If you want to email me to set up an email alert or try to get any of the data, that's here too. And we're going to add to that. We have a user guide that we want to link in here. So this panel will be updated shortly. So now I'm going to go through some case studies, and I'll just go through 4. I'll go through in chronological order. I think I mentioned this earlier, but in 2015 and 2016 in Florida Bay, there was quite a high level of seagrass die-off, and we can kind of see the reason that happened, some of the reasons behind that from these 2 panels. And you can see here, this is 2014, this is July, all the way through the end of 2015. You can see the salinities were quite elevated. Once again, you know that this is the climate. These are the normal conditions here, and then you saw some elevated salinities. It got really bad here in the summer of 2015 when we got salinities up to 43, 45. During that same time, temperatures were elevated, certainly in the summer months. Some cold periods here in the winter time, which is normal. But overall, in 2015, it was definitely warmer than normal water temperatures. And the reason we can see that, and the reason that happened is we can see that in the discharge record, is if you look at the summers, like here's the summer of 2014, normally our when a rainy season is in the winter time and in the summertime and early fall. And you see that here in 2014, and you see it here in 2016. But in the summer of 2015, you just don't see it. We had a dry summer, and there was very little outflow from the Everglades into Florida Bay. And so that basically starved that region of water. And so it was became very saline, and the seagrasses really suffered. So that was visible in this particular record. So that's our first case study. The second case study I mentioned earlier, that was kind of how we all got started on this. And this was the 2016 mortality event at Flower Garden Banks. And you can see this was in July 25th was the observation of it. And this is the discharge record from the 5 Texas rivers here at the top, and then below is the Mississippi discharge record. And you can see, particularly for the Texas rivers, there's a large pulse of discharge in late May until June and early July, and that really caused a buildup of high chlorophyll, high nutrient water, rich in organic matter that built up along the coastline. And you can see that here in the image panels. these are anomalies of chlorophyll A starting in mid-June, running all the way through mid-July. And you can see what happened is we had this large, due to all the discharge, there's a delay of a couple weeks, but basically it all kind of built up along the coast. And so this is, these are anomalies, remember. So this is definitely, this is not normal conditions by any means. And then basically what happened is it got advected offshore. And so, and it's a little hard to see here because this is small. I have another image, but basically the East Flower Garden Bank location is here and the west one's right next door. But the east one was subjected to more of this plume of highly colored water. And we kind of broke that down. We wrote a paper with a colleague from AOML, and we kind of dove into it and got some daily images from other sensors. We looked at Otis. There's actually two Otis sensors. We looked at the other one called Terra, and here on the 22nd, we see that this is this rich, nutrient-rich water offshore here, and it's really going over the East Flower Garden Bank, but the West Flower Garden Bank is a little bit less impacted because this came from the east. And you can see that there was these buoys are no longer there, but at the time there were two buoys over each reef location. And you can see this is the salinity record. The red is at the eastern location, which was impacted, and the blue was at the western location. And you can see here in July, right before this event was observed by, first of all, by recreational divers, and then after that by sanctuary staff, that we had this initial pulse, but then we had definitely a second pulse of inshore water with this low salinity signal that impacted the eastern location, while the western location had higher salinities and less coastal organic rich water. So that's number two. Case study number three is Hurricane Irma, and this was in September of 2017. It passed over Cujo Key here on September 10th, and then came north. And so basically this here is the chlorophyll A, and this is the red reflectance. That's that remote sensing reflectance at 667. And so you can see really it just stirred everything up. And so the path was here where this black line is. And we can see that in the discharge record down here, where we had, the initial event was, we had some rains, and then this is the actual event here with a little bit of a delay. And then, but notice what's really interesting here is that it took a long time. It took almost until, early 2018 for this big pulse that we saw from the hurricane of rainfall to kind of drain out. So there's definitely a delay with these, with these high rainfall events, but they definitely do link to signatures that you can see with the satellite data, but often there is a time lag. And the last case study is the harmful algal blooms that I mentioned. That was in 2018. It started much earlier. These images are from the fall. It started in April and May, and then August was we saw a lot of impacts up here on the beaches. And so these are basically when you can see it when it's at the surface using this algal bloom index product. You can see them at the surface with this product, and like I mentioned earlier, they kind of make it down this way. They weren't observed too much in Florida Bay here, but this is due to the fact that this is really shallow, and so a lot of the times the satellite algorithms don't perform very well here in Florida Bay. But what you do see is that it did make it around the reef tract. It got advected into the Gulf Stream here, made it all the way around, and then in early October, late September, they did actually observe K-Brev cell counts way above normal here on the east coast of Florida. So you can see that in this set of images. And so these are taken straight from the dashboard. They are, like I said, they're done every 7 days. And so that is the case study number 4. And so just to kind of wrap up here, some future plans that we have. Like I mentioned, we want to host these products with GCOOS. They have a great site. They host a lot of different data sets right now. So we want to add these to that. We want to add Insitu water quality data, particularly from the Florida Keys. There's currently there are several data sets taken by different organizations, NOAA, AOML, or Florida Tech, Florida International, all these kind of disparate data sets we want to pull together and try to show either in this interface or something similar to it, and allow users to not only see when and where the data exist, but some idea of what the trends are. Do they match what we see in the satellite? Questions like that are the things we want to get after. And then, like I mentioned, also we want to add some clever animations to show how the coral disease spread down the reef tract starting in 2014. And then, like Chris mentioned in the intro, this is very early on, but we want to, as part of the Marine Biodiversity Observation Network and working with our partners to try to understand what's happening on the biological side by incorporating biological data into interfaces like this. But one of the issues that we find is that biological data is fairly complex in terms of how it's collected. There's a lot of metadata that's included, and it doesn't lend itself to being incorporated into standardized forms very easily. So we're working with that to get it into a form called the Darwin Core and into databases like Otis, the Ocean Biodiversity Information System, and then the end goal would be to serve it in some sort of way that could be visualized, working with partners like GCOOS. So that's kind of where we're headed in the future. And with that, I'd like to wrap up. I just want to thank my co-investigators, our funders. We have quite a few partners on this, people from the Flower Garden Banks and the Florida Keys National Marine Sanctuaries, folks from GCOOS. So this has been a collaborative effort, and we hope to push these into the future, try to get more people interested in using these products. And so I encourage you to reach out to me if you have questions about what you'd like to see, or if you have ideas. I'd love to hear them. So with that, I'm going to wrap up, and I will do a quick live demo. And then maybe while I'm doing that, Chris, if there's questions from the chat board, you can shout them at me. And I'm just going to go through one of these dashboards.
Great. If anybody has questions, if you can type them into the chat box, that would be great.
So while Chris is doing that, I'm just going to quickly scroll through the, this is the FWC Coral Disease Dashboard. You saw this map earlier. So here, like I mentioned, this is where we would select different locations. Let's select the St. Lucie River Inlet. And for our buoy, let's keep it, so we can keep that Peterson Key for now. The reason I choose Peterson Key is because it's this number 3, and it's closest to the reef tract. It would be great if there were some buoys out here. There are some, and they're measuring basically meteorological parameters. But no actual in situ salinity that's being measured out on the reef tract. So this is the map, like I mentioned. These are our satellite image panels. If you click on these, basically it will open that image into a new window so you can see it. This is right now in picture form. You could download it as a picture. And there's a color bar, and it tells you when that image was, what those dates, that these are 7-day weekly means. And so, here are the other products, the algal bloom index. And I should mention too that we would select our time window here. This currently is set for the last 2 years. This is the cumulative sum of disease observations. These as well, you could blow up. Most of these things, if you click on them, you can kind of zoom in to greater detail. So these are the coral disease observations from 2014, when we had roughly 15% of the sites affected. This is a different data set. So these are general, not necessarily stony coral tissue loss disease, but any coral disease that was observed. So right now, we're working with those folks at FWC to parse those data sets so we can try to isolate the disease itself, the stony coral tissue loss disease. Let's see. So this is the satellite time series. And one thing to notice here, if you zoom in closely, you'll see where my cursor is, there's a red line. And that red line appears in all the time series. And so that's handy if you're looking at, say, sea surface temperature, and you find an area, a time period where you want to look at what might have been happening with other parameters, like was it turbid, was there high levels of chlorophyll, what was going on with the discharge, that sort of a thing. So if you notice, if I move my cursor around where we saw this, for example, here at the St. Lucie River Inlet, and it tells you, this is on October 30th, 2019, the chlorophyll spiked way up. And then if you look down here at that particular time, we did see a little bit of a bump here earlier in the river discharge, or in this case, it's the gauge height. It's funny, the USGS measures gauge height on the east side of Florida and discharge in meters cubed per second on the west side, which is, I've always found interesting. But and then these last two are the in situ salinity and the in situ temperature. These are a slightly different look than what you saw before, and that's because we just updated the version of this software we're using. So things, the data is the same, but we're basically have changed the look of it just slightly. And then finally down here, you can get to the data itself. So you can get the ocean color data. You can get the TIFF images of the VIIRS satellite data, and then you can get the time series as CSV files as well. So, so that's kind of just a quick live demo, and I don't know, Chris, are there any questions?
There are. Thanks for that great presentation, Dan. We've got a comment here first. It's always nice to see applications of the ocean observing system data. So kudos to you for that. And then Julie asks, is there any plan to use the model forecast data and compare that to actual data?
No, well, I don't have any firm plans for that, but this sort of interface lends itself to gridded, rasterized model data. I don't know what the outputs are for that, but I'd definitely be interested to hear more.
And I think that's part of the advantage of hosting it on some place like GCOOS, to get that exposure where people recognize those capabilities and can then take it and extend the use of the information. I also had a question for you. When you talked about the regions of interest, so they aim to catch the anomalous conditions along the coast before they actually make it to the reef. So just knowing that is one thing. Are there any plans or actions for mitigation to protect the reefs once these threats are identified?
Well, that's a great question, and that's a little bit beyond, I wish I had the power to affect some of those things, but I can't say that I'm in a great position to answer that question. I would hope that, I think certainly in the Florida Keys, there's been a lot of, increased development, which has caused the reefs to endure higher nutrient loads. But as we're seeing now, you look at the discharge record for the Flower Garden Banks, and we had Hurricane Harvey, and we've had these slow moving, big storms, like we just had one there, Beta, just last week. And so I think we're going to see more of these big, slow moving, high rainfall events where, you know, there are going to be high levels of organic rich waters building up in the coastal regions. And I don't know what you would do to mitigate that, to be honest.
So one of the reasons I asked that question is that there is the National Academies just had an interesting webinar on sustained funding. It was a 3-day event, and the question came back to monitoring not necessarily being a sexy endeavor for somebody to undertake, but that the management application. So you have great monitoring data. Now, how does that translate into our management decisions? And I think that's the bridge that we need to really connect.
Yeah, that's a great question. And, you know, I think it's definitely, you know, you're right. You know, the monitoring's gotten better. We have a lot more technology that we can use now. But what is the end result? How can we improve our lives and the lives of organisms in the sea using that knowledge and the technology that we've developed? But I don't personally have any answers for that. I wish I did. But,
and I threw that out there for the collective, for all of us to think about. It's not something I certainly expected you to answer definitively, but something we all should be thinking about.
Chris, this is Enrique. This is a great point, and I just wanted to chime in about what you just said. And I think that unless we might not be able to come up with a specific strategy to mitigate the effects of freshwater discharge on the reef and that kind of thing, or, you know, extremely warm conditions, this type of data and tools to actually understand what's going on in the environment may help guide research efforts to understand the impacts of these specific events on these different habitats, which is something that is still not very clear. And with better understanding of what's going on with freshwater discharge and how this relates to possible diseases on the coral reefs and that kind of thing, that's when we really can start having an impact on decision making, really. So this is I would say, a long-term process. This is not like, oh, there's a massive discharge going on here, let's go tackle that right away. This is for long-term understanding of these processes, in my opinion.
Thanks, Enrique. That's helpful. Any other thoughts or comments or questions for Dan? Hearing none, thanks again, Dan, for that really informative presentation. And if anybody has any questions, we'll be posting the presentation to the GCOOS YouTube site, and Dan is available to answer questions as well. Thanks, everybody.
Yeah, don't hesitate to email me. Thanks, everyone.